Thank you for the invitation to Tatiana, as well as Aldo and Wayne. Thanks so much. So what I'm going to try and do is a little bit more staying on the conceptual rather than diving into the detail. The broad thing I'm going to try and get to is how you go from kind of a mess of recurrent networks that might be in the middle of the brain, how you go from there to maybe observing something that looks like low-dimensional dynamics within this kind of picture, with the help of course, the slided ones won't work because I haven't wired that up. Let's see if I can get this to work. There we go. How we can go from there to maybe gleaning something like understanding with the help of things like the various omics things that you've heard already, connectomics, transcriptomics, and so on, right? So the general idea for the talk then is something like this. So hopefully you've encountered this phrase before, but if not, it's menu of the day. So this is, to start with, what is a traditional view maybe of what we're after? So this maybe gets to Ken's repeated questions about understanding. Then maybe a starting point that many of us, at least typically in systems neuroscience, have, which is some kind of representation. You do a task, the animal's running around, something like that, and you measure some activity, you have correlations, you have neural dynamics, and that's sort of your starting point in a way. And then getting to mechanism through a path that includes some kind of theory and modeling component, which is how we favor doing things, kind of in a top-down way. How multi-omics can help you test some of the ideas coming out of theories and modeling, to get to mechanistic understanding. And then maybe I'll leave with a little challenge, which is the challenges for, as I see them, for really large-scale models of the brain, okay? So that's kind of the scope of what I'm after. It's going to stay conceptual, and in that spirit, this is giving you my bias. I came to neuroscience as an engineer and used to work on things very different from what I do these days. But my sense of what understanding means is maybe shaped by this background. So to me, it's not just prediction. Prediction is certainly important, but it's also knowing what different components are there for, what computations they perform, what role they perform, understanding the parts enough to know the logic of what the brain is actually after. In this case, the animal is a flying tomcat, which is the other name for an F-14, in case you didn't know that. In neuroscience, this sort of understanding is exceedingly hard to get. So there are a couple of people who were early influences for me at Brandeis when I was still working in a company and taking courses. But for me, this left a mark. So the stomatogastric ganglion may not seem like the whole brain or anything. It's the stomach of a crustacean. But in these 29 to 30 neurons, which have a certain set of types and are interconnected with gap junctions and chemical synapses, they have pretty complete knowledge of a lot of things. And with that, they could, in both conceptual terms as well as mechanistic terms, try and understand how this little circuit generates a variety of different rhythms that allow this animal to grind and process food. And so for me, understanding this link between the smaller scale stuff and then getting to dynamics, well, this small network was kind of influential for how I think of these things. Of course, what I'm after is doing this in larger networks. And so ideally, we'd be able to have this level of explanation where you go from parts to how they tie together, synaptic rules, et cetera, in context where animals are behaving flexibly and you have brains that are far more complicated and generating things like maybe things like this, like a place cell or a grid cell or a head direction cell. And so typically the challenge there is if you go into the middle of the brain, and this is Jeff Tobby's view of the head direction circuitry, if you go into the middle of the brain and you have some densely connected network in there, whether that happens to be a real brain or an artificial one, even if you have some sense of what the task space looks like for an animal or for the agent, what you find in the middle of the brain is kind of messy data. You find responses that give you, correlations to lots of different variables. And so often the terms you tend to hear when describing these response patterns in different areas of the brain that are in the middle of the brain, typically in dense recurrent networks, is things like conjunctive coding or mixed selectivity or notions of, or not everything, sorry, there's a Y missing there, everything, everywhere. And then if you do the right kinds of analysis, or you maybe are biased by, let's say, this is the kind of parameter in a task that you're trying to cater to, or a sensory variable, you can pull out manifolds. In many ways, this is where I began my career in neuroscience. We were looking at how different intensities of an odorant could produce similar response properties. As you change the concentration of an odor, something stayed consistent. if you presented a different order, something changed. How did populations of neurons encode that? We could pull out low-dimensional structure, but in many ways, what I was after ultimately, and the reason I partly moved to the fly, was we wanted to know how do these representations actually arise? What are the generating properties of these? And how does the structure of circuits relate to the dynamics that the circuit produces? To me, that constituted something closer to what I saw in the STG, that was kind of the draw of the fly. More recently, as Srini's talked to you already about, there have been efforts to get the connectome of the fly. So I'd say in many ways, Davi Bok started this at Genelia that led to the flywire reconstruction and segmentation work done there. And in the case of the hemibrain, which followed using a different technique called the FIBSEM, basically allowed us to get the wiring diagram for a large chunk of the fly brain, and soon not just the brain and the ventral nerve cord, but both of them connected together. So these things have been pretty revolutionary. In our particular case, we're interested in a small piece of this puzzle. So the specific connectome that we'll be considering today is for a small brain region called the central complex. It's common to all insects. It happens to go beyond insects as well, to crustaceans. But basically, there's various substructures in this region. There's something donut-shaped called the ellipsoid body. There's this bluish thing called the fan-shaped body. There's this thing called the protocerebral bridge. There's other structures. The names, as Srini said as well, don't matter. Don't get hung up on those. But what I want you to take away is just that this is a small piece of a very large brain. There's certain kinds of topography you'll encounter here, and the structures have very particular shapes that turned out to be quite helpful. So what are we doing there? Well, we're trying to understand how different cell types work together to give you some kind of interesting computation. And we want to, again, the bar for us is like we're trying to understand the computation. But it turns out these different neuron types, and these neurons are distinguished not just, as you'll see, not just by their morphology and their projection pattern, but also by their expression profile. So they are really cell types in some sense. I want to make a little general point here. In the fly, indeed, as Srini said, there is a lot of stereotypy, which is very, very helpful in connecting across animals' behavior, et cetera, et cetera. But a lot of the lessons, for me, from the fly do port to systems where it's not as stereotyped. And we'll come back to that maybe in discussion later. OK, so if you do take cell types into consideration, and you take something like this kind of dense recurrent network, if I told you that this is literally the connectivity matrix all-to-all between these cell types, you'd say, OK, I mean, it looks kind of like there's some structure in there, but fine, it's a dense recurrent network. So if I look at synaptic weights, just assume these are synaptic weights. They're actually relative weights, so it doesn't matter. It's sort of the synapse count between these different neurons. So it's each pixel in this case is the synapse representing the set of synapses between a presynaptic population of, a single presynaptic cell and a single postsynaptic cell. So they're arranged kind of like this at random. But if Dan were to rearrange them with the cell type information that we had, what you get is something like this. So I hope you'll all agree that your visual systems will tell you that this is pretty structured. It's kind of nice. Clearly, there's some reason for this kind of structure. And as you'll see, that makes a huge difference to how you interpret the activity of these neurons as well. So if we were to take just one subpopulation of this, so if we were just to take these neurons that go by the name of EPG and happen to be the ones that are labeled in pink here, if we were to take just those guys and we were to watch them as Johannes did in a fly that's behaving, so in this case in pitch darkness with its eyes painted black, these are the neurons. They happen to project beautifully in these little wedge-shaped things inside this donut. If we let the fly just do its thing and we track in blue the accumulated rotation of the ball it's walking on, and in red the population vector average, so basically the position of this bump, what you get is something like this. So hopefully you'll agree it's pretty remarkable that in darkness it can keep track of its heading essentially as it's walking around. So that's all very well. If we were to take away the topography and we just randomized that entire set of neurons, so you didn't know the identity, you just knew the activity, the time series activity pattern, and you did PCA on that activity pattern, this is what you would see. So this is not just about topography. It's helpful as heck, especially when you're trying to figure out the circuit and how it works. But it's not just about topography. If I just took those wedges and I scrambled them and I did PCA, I would realize that the activity lay on a manifold of sorts, and it's basically like a ring-shaped structure. So clearly, there's something here in the dynamics that is low-dimensional as well, as you might expect. this sort of thing, in combination with just the fact that it moves around in darkness with self-motion cues, there's also the fact that the activity persists in darkness if the fly happens to be standing still. So it's not just about when it moves around, it's that when it stops and restarts, the activity is in the right place. This sort of thing has typically been modeled using ring attractor networks, which have been used in a variety of different contexts, in both mammalian brains as well as fish brains and so on in different ways. But basically, there's a huge body of theory that has put forward quite distinctly different ideas for how different numbers of rings and different connections between the neurons would give you the kinds of activity pattern, activity dynamics that you see here. And some of them have been theorized as lying in this kind of little arrangement purely because that was maybe an easy way to depict things. It just so turns out in the fly, it's sort of literally what happens. So one of the things that I want to say here is, so our approach has been to start kind of simple. So we start with something that just sort of doesn't have every cell type that we know exists. We start with something simpler, and then we build in complexity as in this top-down kind of way, we encounter hitches in our model. So we say we have a simple thing, it recapitulates most features of the dynamics, something's left over, then maybe we add in complexity. So it's kind of the opposite complementary way to what Srini described. It's sort of like taking an approach where you build up things as you go. And so that one representative of that is like Marcella Norman's work in Anne Hermsted's lab along those lines. So let's take the theory-inspired lens onto the connectome. So you have all this structure. You saw that in the early figure that I showed you as well. There's tons of structure. These are each pixel again is the weight between, effectively a weight between one set of neurons and another. These are the compass neurons that happen to be connected to each other. These are various inputs they get. Now, it turns out that there's a very specific kind of model that was out there in the literature from theory that had to do with connecting different types of neurons that would have to have suitable polarity, suitable synaptic connections, and suitable wiring in order to achieve this kind of ring attractor network. And so we could get, in this case, the polarity information, like what are the neurotransmitters, what are the receptors, et cetera, by just looking at these specific neurons that happen to have a particular morphology, which we'll get to what the morphology is in a second. So cholinergic neurons, glutamatergic neurons, okay, there's some excitation in the circuit, there's some inhibition in the circuit. We can kind of ask if the receptor profile matches on the other end, and indeed it sort of worked out okay for us. I won't go into the details, ask me if you like. And what the specific kinds of neurons we were interested in were ones, these were the so-called EPG or compass neurons that projected up into a structure called the protocerebral bridge. So basically, if you think of that little bump that you saw, that little blob of activity that you saw moving around, that got projected up to two parts of this structure, symmetric structure that look like this. So there's two bumps now. Turns out there's another population of neurons that project back, but with a phase shift. What do I mean by a phase shift? If we mark this circle based on what we see with the bump moving around as 0 all the way back to 360, like this, if something goes up from this 180 area, there's a phase shift between these two bits, but we'll ignore that for now. You come back down, and these neurons that bring the information back down project to the neighboring slices of this donut, okay? So far, so good. So you have neurons that take the information up of this bump of activity. When you come back down, you have a phase shift. The notion was, well, if these guys, these other neurons, happen to carry, in addition to heading information about the fly's angular velocity, this would be pretty convenient to shift the bump in one way or the other. So essentially like bicycle handlebars, one thing would move up or move down. If you had a combination of not just angular velocity, but head direction, you could basically shift. If you had input from these neurons back to the original ones, you could shift the bump using one of these two bumps, like basically by just scaling up one or the other. Does that make sense so far? Yes. Okay, so this is something that we could test. So Stephanie Wigner in the lab basically did these experiments where she patched neurons while the fly was walking in darkness and patched from either one side or the other, these neurons that return back to the ellipsoid body. And what she found when she did that was that indeed they had tuning curves to angular velocity that basically matched what we would expect based on these kinds of models. Not a perfect match. In some cases, you might say, why all this variability? Like, why does it go up and down at the edges so much? Turns out, as I told you, it shouldn't just be angular velocity that they're coding. They should also code heading. So if you look at the conjunctive coding of both heading and velocity, then this is what you see. So it's not just the angular velocity, which of course there is a tuning to, but it's also tuned to heading. It gets a combined thing there, projects back, and then so selectively for the bump being over here, these guys would move the bump one way or the other way. Does that basic idea make sense? So you have crisscrossing, you have a combinatorial thing here, and then that's what you get. So in this case, yes, if you didn't know neural identity and you were just sampling from a bunch of neurons, you would see conjunctive coding, but it has a very specific meaning in the case of this computation. It's actually helping to shift the bump around in a certain way. So there's a logic to this circuit that becomes clear if you have access to the cell type, you kind of have a clue what the whole thing is about. So of course, this thing tethers not just to the self-motion, it doesn't just obey the self-motion cues, it actually looks at, it also moves relative to the world around the fly. So otherwise, this wouldn't be very helpful. So indeed, the head direction system here relies on visual as well as mechanosensory cues and tethers to them as an animal moves around. And so as you can see here, it tethers quite perfectly to this pretty arbitrary looking scene. Question is how? So it turns out there's plasticity in the system. And so for the fly to be able to function in the real world where there's all kinds of local and distal cues, and it has to kind of keep its orientation relative to that, what matters, going back to the matrix again, is there's a series of connections going from a variety of different kinds of sensory information carrying neurons onto the compass neurons. So each of these blocks then is basically like one class of sensory neurons. So in this case, they're not sensory in the sense of early sensory, but processed neurons, information that's carried by them is processed. So in this case, let's say we take these specific group of neurons. This is like a visual feature that in some ways encodes large bars, so dark and light spots, but large ones that have that allow you to get a beautiful, exquisite sense of what angle you're at in theory. And each of these neurons, which are called ring neurons, makes synapses. Each of them makes synapses with all of these compass neurons. So there's a bunch of about 50 compass neurons all around, each one arborizing in one little piece of this thing. And each ring neuron makes synapses with all of them, okay? So you have an all-to-all matrix. It turns out in this case, some of these compass inputs are visual. So you might have something like this. So you might have, those of you who study the visual system and other creatures are comfortable, presumably, with something like this, an excitatory and inhibitory profile subfield that basically gets that kind of visual feature information into the compass. It turns out these synapses are plastic. I mentioned this before, and the reason this is key is if you wanted to come into a new setting like this room, and you wanted to map, your head direction system needed to map this setting onto your internal representation of space, you would need it to be plastic. So you want all the cues in this room to be encoded in some way, mapped onto your internal head direction system. And so these synapses happen to be plastic. Important point here is this plasticity, the rules are not so trivial. So this is something we're trying to figure out in a collaboration with Yvette Fisher's lab, which again involved Dan Turner-Evans, who's in the room, but basically turns out to involve multiple neuromodulators. So we're starting to look like the stomatogastric ganglion, for those of you who know it, except on steroids, right? So you have like this internal representation of the world. It involves multiple neuromodulators acting in different ways. If you happen to stare at enough at what the profiles, expression profiles look like, what the transcriptomes look like, you end up with like a zoo of different neuromodulators in different parts of the circuit. Thank you. And so what you end up with is a pretty complicated picture already of just a couple of neuron types that interact with synapses that have all kinds of rules that are shaped by neuromodulators that you can kind of derive in what situations those neuromodulators might be active, but it's not trivial, okay? Nonetheless, we can put all those pieces together for this small set of neurons, and what we can end up with is a model that actually does explain how and why a fly might, in this case, Hannah's experiments, in a setting, in a VR setting where it's actually wandering through the world with translation and rotation, how you can get a stable heading despite all the clutter of the scene and so on using the background cues that you have. But this is just a small piece of the circuit. We've just taken like a handful of cell types, and there's like a zoo of these cell types. So to get what we would consider reasonable understanding, we've had to do away with a lot of the complexity. And it's, of course, there's a lot more there, and the circuit can do so much more than we are giving it credit for. We're not saying that it can't do those things. We're just saying we can explain in human interpretable form just a piece by taking this little piece and kind of exhaustively studying that maybe, or exhaustingly studying that, if any of you are. So, but that hard-won insight is only, again, a piece of the puzzle, because this heading vector that's now been constructed Well, I already told you it goes up to this protocerebral bridge. It comes down to a different structure, and these same phase coding things that the colors tell you here actually propagate back down to a different region. And there's a different set of neurons, many different sets of neurons, of course, that project from these regions back to this structure, down to this structure called the fan-shaped body. Further, each of those gets input of all sorts, including self-motion information, except we can dissect these. And us and other labs are actually in the process of dissecting all these different computations that this circuit is capable of by simply going through the grind of like recording from these different cell types and asking, okay, is this angular velocity? Okay, translational velocity. Okay, this kind of, whatever kind of cue that there is. And each of these, it turns out there's a lovely little vector calculator, trigonometry toolbox, if you will, inside this structure that allows you to do vector math with this kind of phase shifting of neuron projection patterns. And what that allows us to do is slowly but steadily, and slowly, I mean only in the fly context, it feels slow. I think relative to most animals, it doesn't feel it's quite rapid, the progress, but start to make sense of all this structure. It's, yes, of course, it's conjunctive. Yes, of course, it's mixed selective. But in this system, you can really dissect out the component parts in ways that make sense. Like you're trying to get the translational vector out of the head direction system, plus other self-motion cues, for example, so you know which direction you're walking in. And we've used this together with behavior and behavior analysis. This is work from Chuntao and An, to try and make sense of goal-directed behavior where you have reinforcement playing a role in which headings you should like and shouldn't like if you're a fly, reminiscent of the work you heard with lasers on the butt, in this case, of the fly. But the fly can be trained to move away, steer away from some headings and steer towards others. And so what the picture that emerges from this sort of work is something where you don't have just a random recurrently connected network that then gets rewired through reinforcement to control the left and right direction of the fly or the straightforward movement of the fly. What you have instead is a fairly modular brain, lots of plasticity localized to specific sites. So the architecture matters, where plasticity happens matters. To get you rapid learning, what you have is a system where you first maybe build this in the idealized situation. You walk around in the world, you build a map of the world, then you get punished, and then voila, you kind of learn on top of those reference frames you develop by walking around. In the real setting, typically, both of the things happen together. You learn where you are while you realize, oh, there's water over there, or maybe there's something ice inside that for a champagne bottle later, I'm not sure. But you kind of pick up your goals as you go, as you're mapping the world and so on and so forth. And so what you realize is it's not this randomly connected network. It's actually a pretty nicely modularized with plasticity network where different components have different roles. And so that really brings me to kind of the final piece of this. I think one of the big lessons we've learned is you can take all this randomly connected stuff, but it's not random. And if you reorganize it by cell type and you use the information you have, there's lots of structure, and the structure has meaning. And so that leaves open for me. This is, yes, these things are all true. It's not untrue. But there's a deeper level one can go to if one has the tools that actually gives you some understanding of why things are the way they are, what the logic of the system actually is, and what the computations are implemented by. Okay. So I'm going to go back to this original picture I started with. Are we anywhere near something like this? This was a human design system, so you could say we cheated and we knew the answer already. This wasn't, and you just had remarkable humans working on it, and so that's why they got answers there. In our case, a whole bunch of labs and a whole bunch of efforts from multiple teams have led to quite a few tools that maybe gives us some clues into how to go end to end with behavior being explainable in different contexts with different subsets of neurons to circuit level, synaptic level, biophysical detail. But it's a long haul. It's still relevant to do all the old-fashioned experiments. It doesn't become irrelevant. Biophysical details are highly important. What happens at the synapses and the plasticity loop is highly important. It's a daunting number of cell types and connectivity. If you think the fly is hard, you're not going to have it any easier in whatever system you like. The problem is severely under constraint. So you need, if you were taking all of the complexity and throwing something at it, you need a lot of data to actually ground anything, because otherwise you end up with a ton of different hypotheses. Very hard to test them all. So for my case, the smaller scale models, the theory experiment loops, using those smaller scale models may not seem as grand in scale as like taking the whole brain and body and everything else. But to us, the kind of understanding that I get, it has been pretty helpful. So with that, I'm going to thank the folks who made that understanding partially possible. So this is the lab. I mentioned quite a bit of work from Brad. We have an effort trying to get into fish with the help of people sitting back there, named Benjamin Jutkowitz and others. And there's a whole bunch of work. I also showed you a lot of it was actually past work. Johannes Lielig was the one who originally discovered the activity that we now call the compass. Stephanie did those electrophysiology recordings, the plasticity work. A lot of it was done with Sang-Soo originally and is now being done with Mark Flitt and Yvette Fisher and Dan Turner Evans, who did some of the other work that I showed you multiple times. So basically, this is work from that benefited with a lot of help from a lot of people. I'll now take your questions. Thanks.